You're watching CTOM TV. Knowledge is power.
So we're going to talk about testing. Obviously, home testing is one of the best options.
Okay.
Antibody testing is the way of testing for antibodies that are unique to. Can you go to the third slide, please?
Oh, sure.
Antibody testing is there we go. Is the best way to test right now for knowing if you've had exposure to the disease.
And can you tell with antibody testing whether you've had which of those 8 strains you've had?
No, it just tells you that you've had exposure to COVID-19.
Okay.
And basically, what they do is they're antibodies that your body produces called IgG and IgM in response to it. And so it's much like a home pregnancy test or blood glucose monitoring test.
Mm-hmm.
It tells you if you have antibodies that are developed to previous infection. And so it typically takes about 14 to 21 days for you to develop these antibodies against COVID-19.
Okay.
But however, people can remain infected for a long period of time after they develop antibodies. And this is unique to COVID-19. So typically when it takes you about, 14 to 21 days to develop antibodies. But when you develop COVID-19, just because you have antibodies, the assumption is that, okay, now you've got antibodies, you're over the infection, your body's gotten rid of it, your antibodies are proof that your body's got rid of the infection. However, this is not the case with COVID-19. And in fact, people are testing positive for the virus, 6 weeks after they've produced antibodies.
Whoa.
So,
Wow. So normally it'd be like 14 days. You start
almost 6 weeks. Yes. So the key here is you get tested. Then if you have antibodies, you wait 6 weeks and get tested with a PCR or sequence-based test to ensure that you don't have the virus anymore. So all of this stay home for 14 days and then you're, virus free and you're not infectious is just misinformation. And this is the reason we have this pandemic right now. So unfortunately, the FDA has stopped allowing self-testing. They won't allow home-based point of contact testing for patients at this time. And that's unfortunate because there's about 5 or 6 companies that are producing these antibody tests. They're very cheap. People can use them in their home. And there's a variety of reasons. The FDA wants to make sure that people aren't going to be assuming they're negative when they could be positive or vice versa. They want to oversee this. I think it's kind of a bad idea, though. I think all testing needs to be done. And I think this is kind of very relevant. So probably the most important type of testing is called sequencing. And so if you look down at your page here and you see this is a coronavirus, this is a composite coronavirus sequences. Now we're missing a bunch of mutations. I think, can you resize that, Michelle?
Yeah, I'm just going to start again here and see.
Go all the way down, all the way down. All the way down, right there.
Yeah.
Yeah. So for some reason we're missing a bunch of mutations here.
Do I need to set it there?
No, let's not play with it.
Oh, too late. I've already played with it. Yeah. I don't know what's happening here.
Okay, just go down to it, please.
Okay. Oh, hang on. 1 second, everybody. Oh, what is that? Why is that happening? There we go. Okay.
Okay, so right here, there's a lot more mutations here, but if you put your mouse, Michelle, could you go to the right and put your mouse over one of those spikes?
Sure.
These are the different positions of the mutations. So this is codon 84 of open reading frame 8. And as you can see, because it's an RNA virus, please go back to it.
Oh, sure. Sorry about that.
As you can see, this is a mutation that occurs in some strains, and it's 2 nucleotides, 28143 to 28145, because RNA viruses require 3 different nucleotides to code for an amino acid. So there's some functional redundancy in amino acid coding. It's not the same as DNA, so you need two different variants there. So sequencing allows you to determine the different strains. So there are two RNA targeting drugs. One's called remdesivir. It targets the proofreading function of coronaviruses. And so it's very effective. We've seen patients who are on ventilators cured within a few days and released from hospital, where they probably would have died without this drug.
Wow.
The Chinese have done clinical trials of it, and they found it's incredibly effective.
That's amazing how quickly it works. Not just that it works, but
and that's because it interferes with the proofreading. So all of a sudden, the coronavirus is producing non-viable offspring. There's another drug called favipiravir. It creates mutations in the coronavirus, and it also affects the polymerase that transcribes the proteins. It's also quite effective. So these drugs really are excellent for treating. They're new drugs. Well, they've been around for a little while, but there's not a lot of them because there hasn't been a lot of interest in coronavirus. So, I mean, if we could have access to these drugs, we could definitely help a lot of people. Also, because coronaviruses require the furin enzyme to cleave off the spike protein so the virus can enter the cell, or the RNA protein can enter the cell. Furin inhibitors are probably going to be quite effective with this.
And those are readily available?
Not yet.
Oh.
Yes. Well, not specifically for coronaviruses.
Okay.
So there's also vaccines in development. We're going to need a multivalent vaccine. It's probably going to be much like seasonal flu, and they're probably going to be changing it every year. Now, this virus is only mutating at about 25 mutations per year, but obviously we have different strains going on. And, we don't know which mutations are going to cause the ability for it to reinfect. We don't know what's going to happen every year. We know this virus has been with us for a while, and it's only recently become highly pathogenic. But I think, we need to really do a lot of sequencing. And I think the sequencing and, tracking, seeing what's happening in the different strains is really going to be an important thing.
Thank you for watching Cetome TV. Subscribe below and stay informed.